So this first slide is, I really like this slide because it kind of gives you the functions of all the various organs. It kind of tells you what is being digested there. So it's a really good slide. So pay attention to this one. And then we look at the activities. We start with what are the activities which occur in the digestive system. First one is ingestion, taking in food. So that occurs in the mouth or oral cavity. Then, when you take in the food, the first thing you do is you chew. So this is mechanical digestion. Now, for you, chewing is obvious. So you know that that's going to be mechanical. But you don't chew the food down to an absolute pasty form. So when the food passes down into the stomach and even when it goes into the intestine, there too the stomach churns, for example. The intestines swish the food back and forth. So there too you have mechanical digestion, okay? So that occurs both in the mouth, the stomach, and the small intestine, where the food is broken down mechanically into smaller particles. Then you have to actually move the food forward, right? So from the mouth, for example, you swallow, so that's how the food goes into the pharynx. From the pharynx, it goes into the esophagus. So this is propulsion from the esophagus, then into the stomach. From the stomach, it will pass into the intestine. So by waves of peristalsis, this goes forward. So you're kind of pushing the food forward. So that's known as propulsion. Along with all this that is happening is you have chemical digestion, where the food is actually chemically broken down, the food groups. And that occurs in the mouth, where enzymes are present, occurs in the stomach, where again certain enzymes are present, and in the small intestine, where again certain enzymes will be present. Then once the food has been broken down into its smallest absorbable particles, absorption needs to occur, because you have to take those nutrients in. The absorption occurs in the small intestine. Carbohydrates, proteins, and water get absorbed into blood vessels, and in some we'll see the absorption is active, and some it's passive. Active meaning requires energy or ATP, and some it does not. Fats get absorbed into the lymphatic system. You remember in the villi, we had those lacteals. That's why they're there, because they absorb fat. And then the undigested food, which is present in the large intestine, is also propelled and finally passed out. So that is defecation, okay? So in digestive activity, if you look at all of this, there are two main activities which occur. One is the secretory activity, which is what causes the chemical digestion, right? So there is secretory activity, and in the secretory activity, you will notice there are enzymes which cause chemical digestion, but we'll also see that certain hormones are liberated, and these hormones act on the enzymes, or that they stimulate the enzymes to do their job. And these hormones also act on the muscle movement or motility. So you can see, for example, mechanical digestion, propulsion, or even defecation. All this is muscle activity that's going on, which is motility. So the two main activities which occur in the digestive system are secretory and motility, or muscle contraction and relaxation, okay? So let's take a look at this short video which shows you all of this.
Smaller particles during mastication. Salivary glands located near the oral cavity secrete saliva, which begins chemical digestion and keeps the food moist. As food is swallowed, the soft palate blocks the upper pharynx to prevent food from entering the nasal cavity, and multiple voluntary muscles in the face, neck, and tongue contract, pushing food particles through the pharynx. The food passes over the epiglottis, which prevents food entry into the respiratory system, and then into the esophagus, which connects the pharynx to the stomach. The one-way movement of the food mass, now called a bolus, is controlled by wave-like involuntary muscle contractions. This movement is known as peristalsis. The bolus now enters the stomach. Folds in the stomach wall, called rugae, allow for expansion as the stomach fills. Stomach cells secrete hydrochloric acid, pepsinogen, and various regulatory hormones that chemically digest the bolus. Muscle muscular contractions in the stomach churn its contents to further break down the bolus and mix it with stomach secretions to form a thick liquid called chyme. Chyme exits the stomach through the pyloric sphincter and enters the small intestine, the major site of nutrient absorption. The small intestine consists of three parts: the duodenum, jejunum, and ileum. Bile from the liver and digestive enzymes from the pancreas empty into the duodenum to aid in digestion. Absorbed nutrients pass from the lumen of the small intestine into blood and lymph. Chyme not absorbed in the small intestine enters the large intestine. As it passes through the cecum and ascending, transverse, descending, and sigmoid colon, water and salts are absorbed and chyme is converted into feces. The rectum stores feces until nervous stimulation initiates the defecation reflex, resulting in elimination through the anal canal.
So now we look at each of these in turn. So let's see the control. First of all, how is this? Yeah.
Why does it take longer for certain foods to break down than others? Is it because of the chemicals in the food?
Fat, if you have a heavy meal and you have fat with it, fat takes a bit longer because fat tends to get sort of stored on the top, like, and it delays food. It's delayed, like when you say digested, you mean that there is a delay in it, in the stomach emptying. You feel a bit full for a while. So fat kind of tends to delay stomach emptying, so it keeps you kind of full for a much longer time because it tends to float on the top of the liquid contents. Okay, because remember, oil always goes to the top, that's why. So let's look at control of digestive activity. So this is where we are going to make use of the 4 layers of the digestive tract. So remember, you had a local nerve plexus, the Meissner's and the myenteric plexus, which was present in the Meissner's in the submucosa, the myenteric plexus present in the muscularis externa. Then in the epithelium, we have cells which were, remember, those enteroendocrine cells which were present in the epithelium. They produce various hormones like gastrin, secretin, cholecystokinin. We'll look at which area produces what. VIP stands for vasoactive intestinal peptide. There are many more hormones, but we'll only look at these 3 more important. And then if you remember, the autonomic nervous system influenced these 2 plexuses, so the autonomic nervous system becomes really important. And here I told you that in the digestive system, it's the parasympathetic system which is very, very important. So vagus belongs to the parasympathetic system, or the parasympathetic system travels with the vagus. So the parasympathetic system here, so you have from the top. And then the central nervous system also plays a role, because you all know sometimes just the thought and the sight of food makes you salivate, or brings about your juices flowing. Okay, so you can see how the central nervous system kind of also activates this. So let's take a look up here. So when you look at this point here, when you start from here, so here's the lumen and you've eaten food. So inside there are internal GI tract stimuli, and they stimulate receptors which may be chemical receptors called chemoreceptors. They may respond to some chemicals present in your food, or osmoreceptors, which means they respond to the tonicity. If something is extremely too much salt in it, then it will be hypertonic. If the food is very dilute, that may be hypotonic. So these are osmoreceptors which are responding to the tonicity. Or mechanoreceptors. These receptors respond just to stretch. For example, you eat food, your stomach will be stretched, and that'll stimulate the mechanoreceptors. These then they'll send impulses to the central nervous system, and they'll also kind of have reflexes which go in a short way they will kind of stimulate the local myenteric and the Meissner's plexus. Whether they go up to the top and it comes down, or they go locally, the end result is that it acts on the smooth muscle or glands. Glands will secrete, the smooth muscle will contract and will push the food forward or cause mechanical digestion. Okay? So you can see everywhere you have either the receptors will stimulate very short action, or they will pass all the way up and come down, in which case they'll involve the central nervous system and the autonomic reflexes, will come down and they will stimulate the smooth muscle or glands, and you end up with the digestive activity. Remember I told you digestive activity was secretion and motility. You can see it can occur locally like this or through a long loop this way. The other part of the long loop could be you needn't eat anything up here, but just external stimuli. I said just the thought of food, or, even when you smell something, if someone was kind of cooking a steak nearby, sometimes you begin to salivate, or you just are thinking of, oh, nice pie or nice piece of cake or something, you begin to think about food. So that external stimuli can stimulate the central nervous system, which will come down. So nothing from here, but this is just purely from the outside, come and stimulate those intrinsic plexuses, and you still get digestive activity. So let's take a look at this question. Where do you think these receptors are present? Very good. In the walls of the GI tract. These receptors can't be in the pons and the medulla. Remember I said that these receptors are the ones, when food gets in here, that stimulates these receptors, and that's how they send them either locally or to the central nervous system. So they have to be in the walls of the GI tract, okay? So all of those receptors are in the walls. Okay. Again, a bit of review. Where do you think the cells producing hormones which act on digestive activity lie? In the epithelium. Remember, cells producing hormones. Remember what we call them, enteroendocrine cells. This is just the same question worded differently. Remember when I asked you the question, what will the epithelium do? Produce mucus, produce enzymes, produce hormones. So these were those enteroendocrine cells, okay? In the muscularis externa, this is muscle. This is not going to produce a hormone. The nerve plexuses will produce neurotransmitters. The epithelium is the one which had those cells, enteroendocrine cells. Okay, another question. What is the action of the nerve plexuses? Both, yes, the nerve plexuses and even those hormones do both. They cause secretion. You saw there where the nerve plexus caused digestive activity. Remember I said digestive activity was secretion and motility, which is muscle activity. So they do both. So nerve plexuses and hormones do both. Now here, the reason I mentioned enzymes or other substances, because you will see that we will be talking about things like mucus, which we already talked of when we said goblet cells. We'll be talking about bile. All of these are not enzymes. These are other. These go in, and bicarbonate, for example. These are those other substances. So that's what the nerve plexuses as well as hormones too will do. So let's begin with the salivary glands. I told you all there were three main salivary glands. I also mentioned that salivary glands were prone to a viral infection which was called mumps. Yes, it's written here. Mumps. It was which is swelling, so and that, you look like a chipmunk because all these salivary glands are inflamed in that. And if it occurs in an adult, what is the side effect that especially adult males? Testes gets affected. it causes what is known as orchitis. So an adult, and that could lead to infertility. So adult males, that's why getting vaccinated becomes very, very important. So anyway, when we look at the salivary glands, look at this parotid gland right in front of the ear. This part of the ear is called the tragus. It has one duct, which you can see here. This duct passes through a muscle, buccinator, and opens opposite the second molar tooth in the upper part of the oral cavity. Then the submandibular and submandibular duct opens when if you lift your tongue up, you can see a fold present. And I think we did that when we were looking at the oral cavity. There's a fold present, mucus fold, which is present on the underside of your tongue. So the sublingual opens by many ducts. Submandibular duct is single, and that also opens into that little fold which is present on the side of the middle. You know, if you lift your tongue up, there's a midline structure which keeps your tongue from going all the way up, which holds the tongue down. That's called the frenulum. On either side, you have those folds, so that's where these ducts open. In the salivary gland, an enzyme is secreted, which is known as salivary amylase. This is important for digestion. It begins the digestion of starch and glycogen. Starch and glycogen are complex carbohydrates, or also known complex carbohydrates, or also what are called polysaccharides. So when you eat pasta or eat bread, rice, that's complex carbohydrate, okay? Starch or glycogen. So this is where digestion begins. There is a little bit of lingual lipase which is produced by the salivary gland, so some amount of fat digestion can occur in the mouth, but for most part, when I'm teaching you, I'm going to say that fat digestion begins in the small intestine, okay? You may read of it, so you might wonder, you know, what's happening. So fat digestion is very, very little. The lingual lipase is very little, so I'm going to kind of discount that. So I'm only going to talk about carbohydrate digestion beginning in the mouth. The pH in the mouth is more or less neutral, so close to 7. So you can see that the salivary amylase acts at a neutral pH. These glands also produce mucus, which is very important to lubricate your mouth. The salivary glands produce an antibacterial substance known as lysozyme, which kind of keeps your oral cavity fresh and, you know, prevents too much bacterial growth. Substances like defensin, nitric oxide are also produced, which also you can see have an antibacterial, antibiotic type of action. So not only is the enzyme important, but all of these other substances are equally important to keep your oral cavity healthy. The parasympathetic, remember, again, coming back to the digestive system, parasympathetic is very important. So parasympathetic system again controls salivary secretion. And here it is 2 cranial nerves. You read in ANP 1, you read about cranial nerves. 7th was facial. What was 9th called? You can't have forgotten so soon. 9th cranial nerve? Very good, glossopharyngeal, yes, 9th and 7th. Remember, the parasympathetic system traveled with 3rd, 7th, 9th, and 10th. So here you have 7th and 9th. So they control salivary secretion. What do you think happens with decreased salivary secretion? Okay, well, half the class got it right. 1 and 2 are correct. Now, here's why. You have increased bacterial activity, which is why, you know, whenever there is decreased salivary secretion, the odor from the mouth increases because of, you know, the bacteria sort of grow, and that's what they produce, the bad odor. You have difficulty swallowing. So, you know, when your mouth is dry, you know how it's so difficult to swallow. Carbohydrate digestion begins here. That means it's got to end somewhere. right? So there is the small intestine where it ends. So it doesn't mean that no carbohydrate digestion is possible, because when I only use the word begins here, that means it is occurring somewhere else. So carbohydrate digestion would still continue to occur in the other place, and only thing is that the other place, which is the small intestine, is now going to be responsible for all of carbohydrate digestion. Okay, so carbohydrate digestion will still occur. So it's only increased bacterial activity and difficulty swallowing. No, that's too far-fetched. No, the carbohydrate digestion occurs in the small intestine. We have enough enzymes to act on carbohydrate digestion in the Next, let's look at the esophagus. So this is the phase of swallowing. So, take a look at this part over here. Read, I'm not going to go through that. Swallowing has two parts, a voluntary part which is in the oral cavity called the buccal phase. So, you can, till it kind of goes to the back of your pharynx, if you put something in your mouth, you can actually spit it out, you can take it out. Once it goes into the pharynx, it becomes a bit, then it's very difficult to get it out. You've got to kind of tickle the back of your throat to regurgitate that. So this part is involuntary, because remember when we saw one of the videos, and you saw the video in the last part, the epiglottis folds over and closes off the larynx when you're swallowing, so that nothing goes into the larynx, and that means breathing is stopped for that time. So you want this to occur really fast. So this next part is involuntary, because if it was voluntary, you might decide to hold your breath for as long as you want, and we wouldn't want that, right? So the second part is involuntary, so it passes from the pharynx into the esophagus, and then from the esophagus it will go into the stomach. And as I said, respiration is inhibited temporarily, so that's why this occurs really, really fast. Then from here, again, take a look at this video. This is really good, which kind of gives you, again, everything. So now we are going to go to look at what happens inside the stomach. So before we do that, let's take a look at an overview of what's happening.
And however well-mannered we are about getting food into our mouths, there's not much decorum about what happens next. Food doesn't simply fall from mouth to stomach. Your brain triggers waves of muscular contractions that squeeze things along your esophagus at around 2 inches per second. The first place it reaches is the stomach. It's really a biological blender, a churning pool of digestive juices to break down food into easily absorbable ingredients. Leaving the stomach, things are already unrecognizable. Bile, produced in the liver, is added to help the breakdown of fats. At all stages, the body is extracting the nourishment it needs from the passing flow. The walls of our intestines are a vast surface of cells that absorb proteins, sugars, and fats into our blood. It's these that will provide the energy we need for life.
So this is just a short thing showing you, how things are kind of. And the first thing it said is, which is very true, is the stomach is like a biological blender, because it's actually, we all don't chew 32 times. So we do a little bit of mechanical digestion in the mouth and then push everything into the stomach. The food is still in very large chunks. So here in the stomach, you have the stomach churning, which is where it's kind of going to break it down into smaller particles. So that's the mechanical digestion. Plus, protein digestion begins here. So we had carbohydrate beginning in the mouth, protein digestion begins here. And as always, neural and hormonal mechanisms will come into play. The neural mechanisms will be those plexuses, the Meissner's and myenteric plexus, and even stimuli which will come from the central nervous system. Hormonal, we will see there is there's a hormone called gastrin, which we will look at, that comes into play. Both cause digestive activity. which is, remember, digestive activity was secretion and muscle contraction. The secretion which occurs from the stomach is gastric juice, which is secreted, and we will see what are all the contents of gastric juice, okay? So let's see what happens. Just by the process of food dropping into the stomach, this results in stretching, stimulates the mechanoreceptors, which will go and stimulate those local nerve plexuses and cause secretion and muscle contraction. Remember, we said mechanoreceptors was one of the things which caused digestive activity. So here you can see mechanical stimulation. Next is chemoreceptors. So whatever you eat, if there's an alkaline pH plus whatever you've eaten, you know, from the mouth, salivary glands producing mucus have alkalized it a little bit. Proteins present in the food, undigested proteins, caffeine and other substances, they stimulate chemoreceptors. These cause secretion of gastrin from G cells, which are those enteroendocrine cells. So these are those enteroendocrine cells which are in the epithelium. So here we had nerve plexus, here now we have hormone. So here neural and hormonal. So next is hormone. So these G cells produce gastrin, which also causes secretion and muscle contraction, which is digestive activity. The activity in the fundus, because the fundus is the highest part, the activity in the fundus is a bit less. So carbohydrate digestion, which starts in the mouth, it will continue in the esophagus because the pH in the esophagus remains about 7. The moment it comes into the stomach, carbohydrate digestion stops because the pH in the stomach is acidic. But carbohydrate digestion can still occur in the fundus, especially when you have a heavy meal. Some of the food may lie in the fundus. The fundus being the highest part, there's very little activity going on there. Acid doesn't reach that. It doesn't have so many parietal cells. So the pH might remain alkaline. As long as it remains alkaline, a little bit of carbohydrate digestion can still continue to occur in the stomach. Okay? Then the sphincters are closed at this point because you do not want this food. You want this food to be thoroughly churned. You want the protein to be digested as much as possible before passing it out. So the gastroesophageal sphincter will relax and allow food to drop into the stomach, but the pyloric sphincter will remain closed as long as the food is being digested. Okay? In the nerve supply of these sphincters, the parasympathetic system relaxes the sphincters and sympathetic system contracts the sphincters. So whilst parasympathetic system causes muscle contraction for the rest of the organ, in the case of the sphincters, it relaxes the sphincters. So during stomach digestion occurring in the stomach, both parasympathetic and sympathetic act together. So you can see parasympathetic is going to cause a muscle to contract and churn the food. Sympathetic is going to keep the sphincter contracted so that food remains in the stomach and digestion can occur. When food needs to go from the stomach into the small intestine, the sympathetic system will stop. Parasympathetic will kind of override it. It will relax the sphincter, the pyloric sphincter, and food will enter into the stomach. Okay? into the small intestine. So let's take a look at what gastric juice contains. We had two cells which we were talking about. We talked about parietal cells and we talked about chief cells. And then, of course, there were the cells which were lining the rest of which were the simple columnar cells. We call them mucous neck cells and so on. These are the cells which were present in the epithelium. We already talked about the enteroendocrine cells. So let's look at these three cells and what they produce. So gastric juice consists of HCl, pepsinogen and intrinsic factor. Parietal cells produce HCl and intrinsic factor. Chief cells produce pepsinogen. The columnar cells produce a bit of mucus so that they can protect the stomach from its autodigestion. You don't want the stomach to be digested by the acid. The need for acid is because one, you need to denature the proteins. Denaturation of proteins is that proteins are unraveled. Their proteins are tightly coiled. When you unravel them, so you kind of make them straight, it's easier to act on them and break the bonds. So that's why you need an acid medium. Plus, the parietal cells produce pepsinogen, which is an inactive form. In order for pepsinogen to get activated, you need an acid medium. That's why HCl is produced. Pepsinogen, when activated by that acid medium, becomes pepsin, responsible for protein digestion. And this is a positive feedback mechanism. That means the more pepsinogen is converted to pepsin, it keeps going back. The loop keeps going on and on and on as long as that acid medium is maintained. So that's how protein digestion can keep continuing. And it breaks the bond, so it breaks proteins into dipeptides, smaller particles. Intrinsic factor, which is produced by the parietal cells, we did this when we were doing the anatomy, very important for vitamin B12, because vitamin B12 is needed for RBC maturation. If B12 is not, if intrinsic factor is not produced, you end up with pernicious anemia. So as this food is churned and the protein is digested, it becomes highly pasty and is known as chyme. Now there are three phases of gastric secretion. Sometimes your stomach might produce juice without any food even entering the stomach. And that is because everything is in your head. Just the thought of a steak or thought of something really, really tasty cake or something will cause that. Next is the digestion which occurs in the stomach, which is the one which I described. When food enters the stomach and you cause gastric acid secretion. And then the third phase of gastric digestion is when it goes into the intestine. Now you want the stomach to stay quiet for a little while, because you don't want the chyme to be coming from the stomach into the intestine all at one time. You want little at a time, so little, about 3 ml comes into the small intestine digests that and pushes it forward. Then the stomach sends another 3 ml. So you want little at a time to come. So the intestinal phase is actually like shutting off the stomach for a while, and then, so that food comes in little 3 stages. Okay, so when we look at phases of gastric secretion, there are 3, as I mentioned earlier. One in the head, that means nothing touches your tongue or palate, so you're just thinking about something or smelling something. This is entirely neural in the way the juices are stimulated. So you can see sight and thought, or even the smell, goes to the cortex or the hypothalamus, comes down the vagus nerve, so this is neural, and then that will come and stimulate gastric secretion. So it's entirely nervous. Okay. The second one, which is the gastric phase, is the one that we did earlier, where you have both neural control through the vagus and local myenteric Meissner's plexus, and also just the presence of food in the stomach stimulates G cells which secrete gastrin. So you have both neural and hormonal causing gastric juice secretion. After this, the food gets into the duodenum, so you get into what is called an intestinal phase. The initial part of the intestinal phase, for an extremely short time, it continues to stimulate gastric secretion. This is a very, very short time, and this is due to the presence of, in the intestine, a hormone is produced which is called intestinal gastrin. So just for a short time, it stimulates gastric juice secretion. But if you remember, I mentioned that you do not want all of the gastric contents, that's chyme, which comes into the duodenum. You don't want all of it to come at once or to keep coming, because otherwise the duodenum will get overwhelmed and it will not be able to produce enough neutralizing bicarbonate or mucus. So therefore, the second part of this intestinal phase is what is known as the enterogastric reflex. And in this enterogastric reflex, what happens is that the parasympathetic system is subdued, the sympathetic system takes over, so the pyloric sphincter is kept closed so that further chyme doesn't come into the duodenum, and because the parasympathetic system is subdued, so gastric juice secretion ceases for a while. And then once the small amount of chyme in the duodenum passes further, this enterogastric reflex will go off, and then again gastric, chyme will continue to come into that, come into the duodenum. So this is a really good chart, so pay special attention to this. Also take a look at some of these inhibitory events which occur, like someone has a loss of appetite due to whatever reason there is, or there is a protective mechanism in the stomach. If there is excessive acidity in the stomach, you do not want G cells to further go and cause more gastric juice secretion, so the G cells are inhibited, as well as the parasympathetic system is now depressed and sympathetic system is activated. So if parasympathetic is depressed, again, no further juice secretion will occur. So you can take a look at this chart. So it's a really, really nice chart to be looking at. Okay. So here, this is just a review of everything that I said. So 3 phases. Cephalic is through nervous, as you can see, either the CNS vagus, which go, and then the local nerve plexus. The gastric phase, which is in the stomach, you have both hormonal and nervous stimulation that occurs. And the intestinal phase, the initial very short excitatory is due to intestinal gastrin. And then you have this inhibitory phase, which is called the enterogastric reflex. Entero for intestine, acting on the stomach. So enterogastric. So vagus and nerve plexuses, which are parasympathetic, are inhibited. Sympathetic is stimulated, which keeps the pyloric sphincter closed. And it also causes secretion of certain hormones from the intestine, secretin, cholecystokinin, and vasoactive intestinal peptide. These hormones inhibit further gastric secretion. So this, you can see, the intestinal phase has both neural and hormonal actions coming into play. So let's take a look at this.
Regulation of stomach secretion is divided into 3 phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach. In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
Now, hydrochloric acid is extremely important. So we've seen 2 of the ways hydrochloric acid is secreted. It's part of gastric juice. The first was neural, through parasympathetic. Second was through a hormone called gastrin, produced by the G cells. But we have a third substance called histamine, which also causes hydrochloric acid secretion. In people who have excess acid secretion, we often give them antihistaminic drugs. You may have heard of them. These are, you usually have the word idine at the end of it. For example, ranitidine is an example. Cimetidine. These are one type of antihistaminic drugs, so they act on certain cells which produce histamine. So what they will do is they will prevent the release of histamine, which means that no longer will, it will kind of bring down the level of gastric acid which is being secreted, and so that helps in cases of people who have too much acid secretion in the stomach. Another thing you may have been told is that whenever you take aspirin or whenever you take what are called non-steroidal anti-inflammatory drugs, NSAIDs as they are called, you're always told take them on a full stomach, do not take them on an empty stomach because it can cause bleeding. The reason is, if you remember, I told you earlier, our stomach, the columnar cells of the stomach produce a lot of mucus, and the mucus has a very forms a very nice lining of the stomach wall, okay? And what happens is that the mucus secretion is increased by a hormone known as prostaglandin. So prostaglandin increases mucus secretion. What aspirin and NSAIDs do is they reduce prostaglandin synthesis. So if prostaglandin synthesis is reduced, automatically mucus secretion decreases. And so what they do is they disrupt the mucosal barrier. When they disrupt the mucosal barrier, acid can go and injure the cells, as well as the Helicobacter pylori, which many of us harbor in our intestines. They can also go in and interfere with the cells and destroy them, and that's how you end up getting ulcers because of aspirin or NSAIDs. Taking it on a full stomach, the food kind of acts like a coating and prevents this from disrupting the mucous barrier. Okay? So let's review some of what we've done. So what brings about gastric juice secretion? All of the above. Yes, you have both hormonal and neural. The neural is through local nerve plexus or through the parasympathetic, which is vagus. Now, everywhere we saw that there was both mechanical digestion and chemical digestion. The chemical digestion in the stomach was because of pepsin, pepsinogen being converted to pepsin. Also, the stomach needs to churn the food and then undergo peristaltic waves to send the food on to the duodenum. So in the stomach, actually, much like the heart, there is a pacemaker which is present in the stomach, low in the body of the stomach. And this kind of sends these peristaltic, it determines how many peristaltic waves there will be. So first the stomach churns the food and kind of breaks it down mechanically to allow pepsinogen to act further. After it has been acted and the food is kind of made up into a pasty substance which is known as chyme, you can't recognize it when it becomes that. That chyme is kind of pushed out from the stomach into the duodenum. And notice how these waves of peristalsis kind of begin in the body and go on to the antrum and then finally to the pyloric sphincter. The sphincter will open because the sympathetic system is depressed and parasympathetic overrides it at this point. And only about 3 ml of chyme is pushed out, and the rest of the food goes back to be further churned and, propelled down. Notice at this point how in the fundus there is very little activity going on. So therefore, that's one of the reasons why, especially when you eat a large meal, the activity in the fundus is way little at that time, because a smaller meal might come down into the body and in the pyloric antrum, and the gastric juices will be able to act on it. When you eat a very, very large meal, part of the meal may be present in the fundus, so little activity is going on there, which is why salivary amylase can still act on. Remember, it needed a alkaline medium in order to act. So salivary amylase can still continue to act. It does act in the esophagus, but it can still act in the fundus till such time that part comes down here when that salivary amylase will be deactivated. Okay, another question. What is the primary function of the sphincters? Very good. So ensure that food, only one-way passage of food and only little at a time, not sort of overwhelm the next succeeding segment of the tract. Then in the small intestine, let us look at some of the events which occur. First, all three food groups are digested here: carbohydrates, fats, and proteins. Second, this is the region where absorption occurs. So this is, its main function is to absorb, which is why you have so many ways to increase the surface area. Gross or macroscopic was those plicae circularis. Then microscopically you had villi, projections, right, to increase the surface area. Then in the villi you had the brush border, which was the microvilli. So you can see how many ways you increase the surface area. Helps for secretion and absorption. So let's see how the events turn out. So acidic chyme is coming from the stomach. It's acidic in nature, enters the duodenum, where it will stimulate the duodenal mucosa and cause intestinal juice secretion. And this intestinal juice contains water, mucus, because you have lots of goblet cells. The mucus comes from the goblet cells as well as the Brunner's glands, which are present in the duodenum. Plus that, if you remember, I told you that brush border or the microvilli, they produce certain enzymes. So brush border enzymes will be liberated. The enzymes produced by the brush border are enzymes like lactase, there may be maltase, sucrase. These are the enzymes which will act on carbohydrates, and then it also produces proteinaceous enzymes like carboxypeptidase, and so I just have place to write peptidase. So you can see, as you see, the word peptidase means acting on proteins. So these enzymes are produced, so they'll act on both carbohydrates and proteins. One interesting thing about lactase, when we are born, we all have this enzyme, lactase. But as we grow up to be adults, they say that about only 15% of Europeans lack lactase. Most of them continue to secrete lactase through the brush border as they become adults. But in Asians, people of Mediterranean origin and Orientals, about 70% of them, the lactase enzyme disappears when they become adults. So that's why they become lactose intolerant. And that's why dairy products cause a lot of problems in them. So that I thought that was something interesting that you might see, which is why, in the grocery dairy section, you have these lactate and things which don't have lactose in them. That is for these people because they cannot digest that. And then that kind of upsets their gastrointestinal tract.
Now, if, like, say, is it because they don't drink milk that they lose that? Or?
No, it's just a racial thing that happens in them. Like, for example, sickle cell anemia is just, in some ways. It's not because they don't drink milk. They don't know till much later that they are intolerant. Okay.
Could it ever be reproduced? Like,
could lactase come back? They can if they see, it's not being produced. So when if they drink milk or have dairy products which has lactase, they don't produce the enzyme. So I can't say. No, yes, I know usually the body adapts, but in this case the enzyme, because in 70% disappears, I don't think that it can come back, which is why they always have to have something where they have to put, those drops or take lactose-free milk and dairy products. Okay. I don't know. I'll do some research and see. Not that I know of that they are able to get back the production of the enzyme just by drinking more milk. It doesn't always happen. Yeah, I know the body does adapt sometimes. Yes, it does, but probably not in this case. Okay. So that was what happens in the intestinal cells. The other thing is the intestinal cells also contain enteroendocrine cells. Remember all of the tract? So this acidic chyme, when it comes, it stimulates the intestinal cells and causes cholecystokinin. And the name itself should give you an indication of what it will do. Coli is for bile, anything to do with bile, we usually use the word coli. Cysto is for the gallbladder, so usually, when you remove the gallbladder, for example, it's called cholecystectomy, right? So that's how it comes, cholecystectomy. So cholecystokinin, you can see it has something to do with bile and something to do with the gallbladder, which will actually tell you what the effects of this hormone will be. So cholecystokinin and secretin are released by the intestinal cells. What these do is they go into the bloodstream, travel all the way to the liver, where they cause increased bile production and transport from the bile. Bile production is increased more by cholecystokinin. Secretin is more for the transport part of it. Then cholecystokinin also causes, because you need that bile, it passes from the liver. Remember, it was being stored in the gallbladder. You need that bile from the gallbladder to come down the bile duct and get into the duodenum. So cholecystokinin causes the gallbladder to contract. So bile will flow from the gallbladder into the cystic duct and bile duct, and then it needs that hepatopancreatic sphincter or the sphincter of Oddi to relax, because that's the only way it will come into the duodenum. So that's what cholecystokinin does. Then both these hormones also go to the pancreas and stimulate the pancreas to produce their enzymes and juice. So cholecystokinin causes the pancreas to produce an enzyme-rich secretion, whereas secretin causes the pancreas to produce a more bicarbonate-rich secretion. So both enzymes and bicarbonate therefore come into the duodenum. So you can see how the acidic chyme entering the duodenum causes the intestinal cells to secrete intestinal juice, causes it to secrete hormones, which go and act on the pancreas, and then the pancreas secrete their juices. And so all of this bile and pancreatic juice reaches the duodenum. Vagus also causes a weak contraction of the gallbladder. Yes?
I have a question. How does the bile get transported when we move the gallbladder out?
Okay, the question was, how does the bile get transported when we remove the gallbladder? And I'll show this to you in the next picture. Okay? So the function of the gallbladder was, and if you remember, we said bile produced by the liver comes down the... So bile produced by the liver comes down the two hepatic ducts, travels to the common hepatic duct, goes through the cystic duct, stored in the gallbladder, concentrated about 50 times. When needed, this comes back like this and comes down, right? So if you remove the gallbladder, when you do a cholecystectomy, all that happens is that the bile is no longer going to be stored in the gallbladder, and no longer is it going to be concentrated. What will happen is bile will flow down here. The amount of bile, the liver kind of adjusts slightly to how much it's going to produce. So the amount of bile that's coming down is all going to remain in the common hepatic duct and the bile duct. And they do dilate to a slight extent. Okay, they don't become so dilated to resemble the gallbladder, but there is slight amount of dilation. So nothing else happens. The only difference is that your bile is more watery. It's no longer concentrated. Okay, so that's so doing a cholecystectomy doesn't really have any adverse effects. So again, I like this picture really a lot. So take a look at how this, everything that we just said, this is what it does. So what is missing here is chyme first causes secretion of intestinal juice as well, and then it causes the enteroendocrine cells to release their hormones. These hormones enter the bloodstream. They go to the pancreas, where cholecystokinin causes an enzyme-rich juice and secretin causes more bicarbonate to be produced. They also go to the liver, where both, here bile salts is written in your chart. You may want to correct it to cholecystokinin. Cholecystokinin and secretin stimulate the liver to produce bile more rapidly. Cholecystokinin also acts on the gallbladder, contracts it, and at the same time relaxes the sphincter so that bile can travel down and get into the duodenum. And because both pancreatic duct and bile duct open through a common opening, when this sphincter is relaxed, you can see that bile will come into the duodenum as well as pancreatic juice will come. This juice has bicarbonate, which will neutralize the acidic chyme, and plus the intestinal juice also has a lot of mucus from the goblet cells as well as Brunner's gland. So that also will help to neutralize the acidic chyme which comes in. Because otherwise, if the chyme is so acidic, it will first burn or completely erode the duodenum. Second is you want the pH to be alkaline here because all of the enzymes act at an alkaline pH. The enzymes produced by the pancreatic juice, they act at an alkaline pH. This is the reason why you have only 3 ml at a time, because the amount which is produced is able to neutralize 3 ml. If you had too much of it coming in, it would not be able to neutralize it. So not only would it cause ulceration of the duodenum, it will also not activate these enzymes which are coming from the pancreas. Okay. So let's look at what bile and pancreatic juices contain and the function. Bile is not an enzyme. It only causes mechanical digestion. It emulsifies fats. That means it breaks them down into smaller particles. You have a big fat globule like this. Lipase, the fat, the enzyme which digests fat, will only be able to act on the surface. However, if you broke this down, which is what bile does, it breaks it down mechanically into smaller particles. So now lipase can act on all of them, all of the particles. So all of the fat is digested. If bile is not produced, or if say there's some problem and bile is not going into the duodenum, you would still have digestion of fats. You would have just digestion on the outside. Most of the fat would not be digested. So some amount of fat digestion will occur. Most of it which is not digested will pass out in the feces. And so, the stools would be extremely greasy and you would lose a lot of fat in the feces. So that's why bile is important. So it also facilitates fat absorption. So you can see not only for digestion but to absorb the fat. Cholesterol is present, is one of the components of bile. So it keeps that cholesterol dissolved in bile, and bile has bile salts and bile pigments. So what it does is it makes sure that the relationship or the ratio between bile salts and cholesterol is kept constant. If that ratio changes somewhat, you get more cholesterol or more bile salts, and the ratio is disturbed, you'll end up with gallstones because it precipitates, and that's when you'll have to remove the gallbladder. It also has this pigment, bilirubin, which actually when it is transported through, this bilirubin is removed in the urine as something called urobilin, which is what gives urine the yellow color, and it's removed in the feces as what is known as stercobilin, which is what gives feces the yellow color. It is this, whenever there is excessive red cell breakdown, the hemoglobin pigment which travels through, that hemoglobin pigment also passes through the liver, and excessive bilirubin is produced, which is what that bilirubin pigment is deposited under the skin and mucous membrane, and that's what gives rise to the yellow discoloration that we see when someone has jaundice. Okay. Let's look at what pancreatic juice contains. It contains all 3 types of enzymes which can act on the food groups. Proteases, you have like carboxypeptidase, trypsin, chymotrypsin, all these are protein digesting enzymes, so they digest proteins. We have pancreatic amylases which digest carbohydrates, and pancreatic lipase which digests fat, and then the bicarbonate ions to neutralize the acidity which is there in the duodenum. And let's take a look at secretin and cholecystokinin and review this.
My lower back pain.
Horrible.
So excruciating.
Welcome to the tutorial. This time we're going to look at how CCK, which is a hormone, causes bile release from the gallbladder. So first of all, a little bit of digestive anatomy. We've got the stomach here, which has the esophagus that leads into it. And coming out is the first section of the small intestine, the duodenum. Off to the side is the liver here, and underneath the gallbladder, and then the bile duct, which leads from the gallbladder into the duodenum. And that's what carries the bile. So over the stomach again, when you have fats in your diet, as we inevitably do, a lot of the lipids are clustered together in sort of large globules, illustrated by this. Whereas single lipids that make up the globule would be represented by just one L here. Now the hormone CCK is going to be involved, and you'll see how in a second it stimulates the release of bile. And bile is going to go in and cut this lipid cluster down, so we have single lipids, and then lipase, the enzyme, will act in the duodenum and break down the lipids for us. So let's watch the animation. So the lipids are in the stomach, they come down and rub the wall of the duodenum. The hormone CCK travels through the blood to the gallbladder. The bile comes out and splits the large lipids into small ones. And now these small lipids can be acted on effectively by lipase. So the action of bile there doing the splitting part is called to emulsifying the lipids. Okay, so I hope this helps you.
So you can see that it doesn't digest, it only helps to emulsify. So this is just mechanical digestion, not chemical digestion. Now, in the small intestine also, when we did right in the beginning, if you remember, we said we had mechanical digestion occurring in the mouth, which was chewing, occurring in the stomach, which was churning, and in the small intestine, it is known as segmentation. The idea is that the intestine is kind of broken up into small segments, and food swishes back and forth between the segments. So can you see how it's kind of swishing back and forth between these segments? So when it's completely churned, it's broken down even further, making it easier for the enzymes to act. After this, the food, once the food is completely digested, then it will start peristalting and get pushed on towards the ileocecal valve. So peristalsis is very different because it's a wave that goes forwards. As you can see here, it's a wave that goes forwards. And true peristalsis only starts after absorption has occurred. So let's take a look at how segmentation occurs in the small intestine. And you'll see here how the food is swishing back and forth. So notice these segments. So it's going back and forth between the two segments. So take a look at these, see how it's going back and forth. So this is, so each part you can see is just swishing it back and forth. So this is, so notice how it's going forwards, then it'll come back into the same segment. So this is very typical segmentation that occurs. Then absorption in the small intestine. So by the time chyme reaches the ileum, the ileocecal valve, most of the absorption has occurred. Water, electrolytes, and all your various nutrients. Carbohydrate and protein absorption is active in nature. That means it utilizes ATP. Fat absorption is passive. Absorption has to be in the simplest form, which means carbohydrates get broken up into glucose, proteins get broken up into amino acids, fats, fatty acids, and glycerol, or what's called monoglycerides, and glycerol. Glucose and amino acids, as you can see here, enter the bloodstream. Fat goes into the lacteals. And here's a nice picture showing you how fat absorption actually occurs. So the fat globule is broken down by bile salts and made into smaller particles, and which is acted upon by the lipase. So when it's digested, it yields free fatty acids and monoglycerides. They can't cross the epithelial lining. So these actually associate themselves with bile salts, and they form what are known as micelles. These micelles are able to pass through and they get into the epithelial cells. Once inside the epithelial cells, they recombine and they become triglycerides again, but this time they get attached to proteins. So the attachment of fats to proteins is known as chylomicrons. These chylomicrons then pass into the lacteals, from where they are taken into the thoracic duct, which is why the lymph in the thoracic duct is known as chyle, because it has a lot of chylomicrons in it, and it is very milky in nature because fat looks a bit milky. And here let's take a look at how this absorption occurs.
The lining of the small intestine is characterized by numerous circular folds called plicae circularis. The plicae are lined with finger-like villi. From a cross-sectional view, the villus contains a network of capillaries which surround a specialized lymphatic vessel known as a lacteal. The epithelium of an intestinal villus consists of columnar cells which are covered with microvilli. This succession of folds and projections increases the surface of the intestinal lining for efficient absorption. Carbohydrate digestion is completed by enzymes in the small intestine. Carbohydrates are absorbed by the villi and then enter the capillary. Fat digestion occurs primarily in the small intestine. Fat molecules are digested and absorbed into the epithelial cells of the villus. The fats are formed into clusters called chylomicrons, which pass into the lacteal. Lymph carries chylomicrons away from the villus. Protein digestion is completed in the small intestine. Proteins are broken down first into peptides, then into amino acids. These are absorbed into the villi, then into the capillary.
This again is a really nice slide for you to review. I mean, I'm not going to go through this slide. Take a look at all of this so you can see carbohydrate digestion. It gives you the various enzymes. Protein digestion, various enzymes. Fat digestion, it mentions lingual lipase and gastric lipase. Small amounts. I've not kind of taken those into account. I've only kind of mentioned the pancreatic lipase. Okay, so I've really not paid too much attention to this because there's a slight amount of digestion by this. Most of it is done by pancreatic lipase. And then we have other substances like nucleic acids and so on, which are also digested. And you can see the brush border enzymes up there for both carbohydrate and fat digestion. Carbohydrate and protein digestion. So pay attention to this. Don't kind of gloss over it. It kind of shows you real well how it is absorbed, where it is absorbed, which enzyme acts on these different food groups. So let's review a little bit. So which of these statements is not true? Very good. Bile does not digest. When we use the word digest, we are talking about chemical digestion. Remember, I said it's an enzyme. It doesn't digest. It just breaks it down, okay? It's lipase which digests fats. Okay, another question. Yes, contraction of the gallbladder and relaxation of the sphincter, not making the pH neutral. This is a hormone. This won't make the pH neutral. Another question. Okay, brush border enzymes are produced by the intestinal epithelium. Remember I said the intestinal, if this was a villus, and this was the cells of the villus, lining the villus, and I take one of these cells, each of these cells on the top has microvilli, which when you look, and if you looked at that video too, you saw that it kind of gives a little brush-like appearance, which is why it's called brush border, okay? So the enzymes are produced by this. The Brunner's glands are way here. They only produce mucus. So it's brush border enzymes. Muscularis mucosa is the muscle layer which is present in the mucosa. Muscle cannot produce enzymes, so this muscle is not producing the enzymes. Next, when we go to the large intestine, its main function is absorption of water from the undigested food that comes in. Some electrolytes are absorbed here, and then what is left behind is feces, and that is eliminated. In the large intestine, I told you, were present those symbiotic bacteria, also called intestinal flora. They produce vitamin K and B complex. This is the reason when this vitamin K, which is produced by this, is enough for all our body needs. So and vitamin K is very important for forming clotting factors. So when children are born, neonates as they are called, if they have to go up for surgery because their gut is sterile, because it hasn't yet been populated or colonized by bacteria, we often have to give them vitamin K injections so that we kind of speed up the formation of clotting factors, because they're going up for surgery, you expect that they'll bleed. I also mentioned that when you put somebody on broad spectrum antibiotics, you end up killing the bacteria you want to kill, but you also kill off these bacteria. And so B complex, you might end up with B complex deficiency, which is why people tend to get mouth sores, and you ask them to take, especially if they are on broad spectrum antibiotics for a long time, you want them to take supplements of B complex, so to kind of make up for this. Final act of defecation, as you can see, is under voluntary control, because the external anal sphincter is a voluntary sphincter. So let's take a look at these probiotics. It's a nice video which shows you the importance.
In most cases, the organisms in our food get mashed around in the mouth, then hit the roller coaster that takes them rapidly into our stomach. The stomach is not a particularly inviting place. Acid is produced in large quantities when we eat. This helps to protect our body and to break down food, and it kills many bacteria that we ingest, including many of the milk fermenters present in dairy products. However, some bacteria, such as these probiotic lactobacilli and bifidobacteria, can survive the acid by having a protective shield, shown here as a halo. This protection can be provided by milk and food, or by leaving the stomach rapidly with the liquid part of the meal. More than half of the world's population, and especially people in Asia, have a bacterium called Helicobacter pylori living in their stomach. Some strains of this bacterium are pathogenic. They're acquired from several sources, including contaminated water. This bacterium can burrow into the walls of the tissue and produce urease and toxin that damages cells, causing painful ulcers and sometimes cancer. Antibiotic treatment can cure some cases of Helicobacter colonization, but in others, the bacteria either resist the killing or return again once the treatment is over. Probiotic bacteria may help in the reduction of these Helicobacter, perhaps by attacking them with antibiotic-like weaponry called bacteriocins, or dislodging them. However, more research is needed to understand this process, while clinical trials will help determine which patients may benefit from ingesting certain types of probiotic bacteria. The dead organisms, along with the probiotic bacteria and surviving fermenter microbes, move towards the intestine, where they come into contact with bile salts, which kill many of the milk fermenter microbes. Many probiotic bacteria can resist these harmful effects, and so they survive and continue their journey. The next stage takes the bacteria down through tissue tunnels into the small intestine. This region of the body is usually over 30 feet or 9 meters in length. It has structures called villi and microvilli, which absorb nutrients from food and form a major defense against disease. The small intestine is home to billions of bacteria, the microbiota, including bifidobacteria and lactobacilli, which play a critical role in keeping us healthy through degrading food, counteracting bad bacteria, and modulating the immune response. Many questions need to be studied, such as what types of bacteria live here, how they communicate with each other and influence the way we feel, as well as whether probiotics can prevent or alleviate certain diseases.
That was so that probiotics do a lot more, as you can see, than, just giving us B complex and vitamin K. Okay, let's look at the fate of everything that we've eaten and digested. And two basic processes occurring in the body. Anabolic process means building up. When you build something from small molecules, become large molecules. So when you build something, you use ATP, right? If I asked you to build a house, you would need energy to do that, okay? So you build that. Usually in whenever there's something called anabolism, you will see the word genesis because genesis or synthesis is linked to that. Because the word genesis means to make. Synthesis also means to make. So you make something. Catabolic process is to break down. Another word for breaking down is lysis. So you will usually see the either breakdown of proteins, for example, or you'll see the word lysis attached to another word, okay, like lipolysis. So that means lipids are being broken down, okay, so that's a catabolic process. Catabolic processes liberate energy. So they liberate ATP because, you're breaking from a big thing, breaking something down, you don't need energy, you actually liberate energy. So in your notes, you have a lot more given, so I would like you to read your notes. I'm not going to kind of go through all of that, but I'm kind of going to, just in brief, tell you what the fate of each of these nutrients is. So read your notes very, very carefully and kind of add to that, because I would just be repeating everything that's there in your notes. So one thing is, when carbohydrates are digested, they have to be broken down into their simplest substances. Those simplest substances are monosaccharides. So they are broken down into glucose and fructose, maybe even galactose. These fructose and galactose are in turn converted into glucose and then absorbed into the bloodstream. The glucose then is the source of ATP production. It's your most immediate source of ATP production. That's why when you're tired, you always take something really sweet, because it gives you immediate energy. ATP is the only utilizable form of energy. So while glucose may be the source, that glucose actually has to be broken down into carbon dioxide, hydrogen, and ATP for your body to be able to utilize it. Now, you don't take just enough carbohydrates to give you the ATP you need for your daily activities, right? You take more than you need. Whatever excess glucose there is taken to the liver and skeletal muscle, where it is stored as glycogen. Glucose molecules are joined together and made into glycogen. So you can see the word glycogenesis. So this would be an anabolic process. It's stored in the liver and skeletal muscle, so that if the body needs, let's say you haven't eaten food for some time, but the body still needs energy, this stored glycogen can then be broken down to provide glucose, which will be used for ATP. So then the glycogen will be broken down, so that's called glycogenolysis. So what will this be, anabolic or catabolic? Catabolic, that's right. Now, you may have heard of athletes. As I said, glucose is a source of energy. The stored glucose is glycogen in liver and skeletal muscle. So, and this, what athletes do is perfectly legal. They do something called carbohydrate loading. So, and you can understand why they would do that. So 2 days before, or 2 or 3 days before their event, like a race or a tennis player, I love tennis, so that's why I always talk a lot about tennis. Tennis players tend to have a big meal of pasta or anything that is carbohydrate rich. So what happens is that carbohydrate goes in, it's whatever glucose is needed by the body is broken. The rest of it goes and gets stored as glycogen in the liver and skeletal muscle. And then when they are, doing their activity, that glycogen can be broken down to produce glucose and give them ATP. So this process of loading your body with carbohydrates is called carbohydrate loading. It's perfectly normal, and it's so nothing illegal like, doing, giving yourself like blood doping, okay? Now you may eat excess carbohydrates, so you've stored your glycogen, but there's still some more glucose left. That glucose goes and makes proteins, and it will also go and make fats, which get stored in adipose tissue. This is the reason if you have a very, very rich carbohydrate diet, you end up storing a lot of it as fat, okay? Proteins are not stored as proteins are broken down into amino acids, but they're not stored as amino acids. The proteins are those amino acids are quickly used up to either produce ATP, or they're used up for growth and repair, or to make other protein substances in the body. Can you tell me some protein substances in the body? Yes? I heard something which sounded familiar. Yes, very good. Enzymes. Amino acids form proteins. They are the building blocks of proteins. But I'm asking you, what are protein-containing substances? Come on, ANP1. In muscle, yes, remember muscle protein. You have, remember troponin, actin, myosin, all that, okay? So muscle, we'll say muscle protein. Anything else which you've actually done in ANP2, I want you to kind of concentrate. Think back right from the first lecture. What's that? Cell membrane are lipoprotein in nature, okay. How about immunoglobulins or gamma globulins, right? Protein, globulin is a protein. Albumin, remember in blood chapter, albumin is a big, very large protein. Of fibrinogen, all those clotting factors, protein. Hemoglobin is a protein, right? See, so many, right from the first chapter. So many things are protein in nature, so that's what it is used for. Any excess protein you may have, that protein can be converted into glucose and then that glucose can be broken down to give you ATP. So that protein from a non-carbohydrate source, remember when we did endocrine system, we talked about it. So when proteins from glucose, they are known as gluconeogenesis. That means the genesis of glucose from new sources. Glucose is a carbohydrate, so it usually should come from carbohydrates. But here you're getting glucose from a new source. And proteins may also be used, if there's excess protein, they may also be, those amino acids may also be used to form lipids. So you can see how each group can actually kind of be converted into the other group. Yeah.
So if you're on people do the no-carb diets and they eat tons and tons of chicken and tons of protein, will that eventually, if you overdo it, that eventually...
Anything which you overdo will harm. You should never go on a diet which is completely eliminates one of the things. So if you have too much of that, you feel excessively hungry, you may end up to the process where you may form lipids. You may end up. The only difference is that proteins are very filling, so it's very difficult to eat too much of it. Lipids, when lipids are broken down, they form fatty acids and monoglycerides. These then, remember, get formed into triglycerides combined with proteins, and then as chylomicrons they go into the bloodstream. And then these are taken to various parts of the body and stored over there, and one of which is adipose tissue. You have also other type of fats which you see in your notes. Those are known as very low density lipoproteins, VLDL. Then we have low density lipoproteins and high density lipoproteins. These are different types of fats that you have. Low VLDLs take cholesterol which is formed in your liver and take our fats and take it to adipose tissue for storage. Low density lipoproteins, which are the ones which are called the bad fat. You don't want to have too much of it, because if you have too much of it, it can get deposited in your arteries as plaque, and then that will could cause thrombosis, could raise your blood pressure because of increasing peripheral resistance. High density lipoproteins actually remove the fat from tissues and take it to the liver so that it can be broken down and removed. So the higher your HDL is, that's why these are called good fats, and these are known as bad fats. So the more you have of HDL, the better it is, because it takes fats present and takes it to the liver and removes, and where the liver kind of breaks it down and removes it. Lipids are used, and you need to have some amount of fat in your diet, because cell membranes are lipoproteins. Myelin is a lipid substance made out of lipids. Certain hormones, steroidal hormones, have lipid in their configuration. So you do need lipids for for manufacturing these substances. So that's why some amount of fat should be there in your diet. And then fat is also a very good source of ATP production. And any excess fat there is, that fat is converted into glucose through gluconeogenesis, and then through that route also you could produce ATP. So let's answer this question. Which of the following is a catabolic reaction? Remember the key words that I mentioned, so this really shouldn't be a difficult question to answer. Glycogenolysis. See, the word lysis is attached. This is breakdown. Everything else, your genesis is you're making something. You're making glucose, you're making glycogen, you're making lipids. Here you're breaking down glycogen. So this is a catabolic reaction. Now in metabolism, again, make sure you read your notes. So I've kind of tried to break it down to because metabolism is really a biochemistry chapter. So let's see what, y'all don't have this thing. Okay, this is just, it's not very important because the one after this shows. So here when you look at it, when you have organic molecules like amino acids, lipids, and simple sugars, they are broken down into their respective, these molecules are broken down. They go into their nutrient pool, and here some of it is their catabolism occurs, some of it is used for anabolism. So inside the cytoplasm, this is processed where ATP is formed, and actually only 40% of either one of these makes ATP. Rest of it, 60%, comes out as heat, which is what happens when basal metabolism goes on. This ATP which is produced is used. Why do you need ATP? Because you want to synthesize new molecules for cell maintenance or other functions. For example, when we did active transport in ANP1, like you remember phagocytosis, endocytosis, sodium-potassium pump. So want to use all of that, okay? So here again, so this was what happened in the cytoplasm. When we come to the mitochondria where oxygen is used, you will find that it enters through various cycles, and that's how ATP is produced, and water with carbon dioxide. The water remains in the cell. The carbon dioxide is something that you pass out of the body. So don't worry if you don't have this. Let's look at the same thing up here when you look at stages of metabolism. So in the first one, digestion in the most absorbable form. So proteins are broken down into amino acids, carbohydrates into glucose and other sugars which will be converted into glucose. Fats are broken down into glycerol and fatty acids and then converted into fats again. So glucose enters first into in the first phase. So this was all this where amino acids make proteins, glucose and sugars make glycogen, glycerol and fatty acids combine to make fats. That is anabolism. So that part goes into the forming intermediates within cell tissues. Otherwise, when you get glucose, glucose first enters a cycle which is known as glycolysis. Whenever you need energy really fast, you don't have the time to take your time and get oxygen and then produce ATP. So when you need energy really fast, it undergoes a process without oxygen which is known as glycolysis. The end result is pyruvic acid, and you usually end up forming about 2 ATP. So this process of glycolysis, as you can see from this chart, can be through glucose, it can be through amino acids, or it can be through glycerol. Okay, so only 2 ATP is produced, but it occurs really fast. So this, the body will use, for example, let's say you want to run a 100-meter sprint, which just takes a few seconds. You'll use this form of energy. Then some of the amino acids could go into forming a second coenzyme, which is called acetyl-CoA, and fatty acids also could go into acetyl-CoA, and it goes into another cycle where you need ATP to be generated for a longer period of time, and you need oxygen for that. So this is called where oxygen is needed. So it goes through one cycle which is called Krebs cycle, and this Krebs cycle kind of liberates hydrogen ions which have energy in their bonds. These end up going into something known as the electron transport chain, and lot of ATP is formed at this point. So you can see that up here you have less ATP formed, but it's very, very quick. When you go into what is called the oxidative phosphorylation, which is through a chain of electrons which have coenzymes and hydrogen keeps getting attached to it, hydrogen has energy in its bonds, then this takes a while, but you produce a lot more ATP. So somewhere, let's say you want to run a marathon, you would use this kind of energy producing mechanism. If you want to run the 100-meter sprint, you would use this kind of energy production. Okay? Now in the mitochondria, again, like I said, this is a lot of biochemistry really, so we don't do it too much. So there are two sort of processes, one which occurs in the cytoplasm not requiring oxygen, so that is glycolysis. You produce only 2 molecules of ATP, and you also produce pyruvic acid. That pyruvic acid goes into a part of the cycle called Krebs cycle, and from there it goes into the electron transport chain. In the Krebs cycle too, actually you form about 2 ATP molecules, small amounts, and then once it goes into this electron transport chain, this is the one where you have a lot of ATP generated, somewhere like about 34 molecules of ATP. So this is kind of, I really want you to know where glycolysis occurs, which is in the cytoplasm. Krebs cycle and electron transport occurs in the mitochondria. Both glycolysis and Krebs cycle produce small amounts of ATP, whereas the electron transport chain is the one which produces large quantities of ATP. And most of the ATP is what is produced by this. So let's take a look at this last question. This person has not eaten for 6 days. So which of the following reactions do you think would not be of importance in providing ATP? So I want some more people to think on this. No? Okay. 40% of you got it right. Protein synthesis is not of importance because you're synthesizing protein, you're making ATP. So let's look at this one. I mean, you need ATP to make proteins. Glycogenolysis. Glycogen is broken down. It will give glucose. Glucose will provide ATP. Gluconeogenesis. Even though this is genesis, remember you're going to produce glucose, which can be broken down to give you ATP. So if someone is starving, for example, they don't have stored glycogen. What are they going to do? They're going to take proteins and fats, make glucose from them so that that glucose can be broken down to give ATP. Lipids can be broken down to provide ATP. So all of these will give you ATP. Remember, we talked of all three of them. Protein synthesis, you will sit and make proteins at this time. You don't want to be making anything at this time. Instead, you want to break down the proteins to provide ATP. Here, you're going to use ATP to make proteins, okay? So when someone, think of this, she's not eaten, she's starving. At this point, muscle, you've seen people who are starved. Don't you see that their muscles actually break down? They look much thinner. That is because proteins are broken down and muscle breakdown occurs. So you're not building muscle at that time. I mean, muscle being one example of where protein synthesis occurs, okay? So this is one way that the body will not provide ATP. All of the others would be useful ways to provide ATP in this situation.